basically cover how does real-time PCR work, what chemicals and instrument detect DNA already or RNA. In fact, ma'am has already discussed a little bit about that thing, and I will be discussing a little bit more. And how real-time, how mainly I shall discuss how real-time data look like and how real-time data can be interpreted and optimized. So I will be taking more this presentation like a more of like a workshop like thing, so that the person who are so following this thing, they can able to interpret in their lab. So before starting, just to see just 5 seconds, I shall spend on why the molecular diagnostic has come an emerging area of science today. First reason is a quicker turnaround time. The improved sensitivity and specificity, accuracy is also very increased, and if you see in a large scale, it is also a cost-saving technology. So for that, the molecular diagnostic market was 8.3 billion in 2019, but I think it will shoot up after the COVID-19 event. Now, before starting the real-time PCR, let's have a look on conventional PCR or polymerase chain reaction as a technique, because if we do not understand conventional PCR, or then it is very difficult for us to understand real-time PCR. So already I have discussed this thing. So let's move on to the next part. Polymerase chain reaction. So if we go to a molecular lab, these are the basic techniques. First, the extraction of nucleic acid and purification. Next step is the amplification, making copies, which is done basically by real-time PCR. Then finally, the detection technique. Detection can be done like an end product detection, that is in a conventional PCR by gel electrophoresis, and it can be also real-time PCR. So real-time PCR is just like a GPS kind of thing. So where we can track the amplification production in real time, in every seconds. So if we see PCR as a technique, it can amplify one gene, several genes, lots of genes within very short time. So what are the requirements of a PCR? First, the template, then pair of primers, forward and reverse. Next, the thermostable DNA polymerase, then DNTPs, then a buffer to maintain the pH, and finally the PCR machine, which is thermal cycler. This is the basic requirement to start a PCR. So template DNA can be any type of DNA or any target DNA. So there is no discrimination that what it can amplify some DNA or it cannot amplify other DNA. So any DNA or any sequence can be amplified, provided we know the sequence a little bit, the two parts of the start and end sequence, so that we can design the primers. Next, we need a pair of primers. If one is called forward, then the other would be called reverse primer, which we would be targeting the genes we want to amplify. Next, the DNA polymerase, but you can understand we cannot use our own DNA polymerase, because in PCR we have to increase the temperature to 90 to 95 degrees centigrade, which temperature our own DNA polymerase cannot experience. So that's why the scientists have extracted DNA polymerase from a bacteria called Thermus aquaticus, and that's why this polymerase is known as stack polymerase. This polymerase works well, does the amplification in the A, T, G, and C. The each magnesium ion the samples, the double strand between and T and triple hydrogen bond between G and C is broken, and once it is broken, the double stranded DNA becomes single stranded. Now, once the DNA becomes single stranded, the denaturation of the DNA, that what should be your denaturation temperature. And next is the hybridization temperature or standard DNA. Now, the choosing hybridization temperature or Elling temperatures are very tricky. Of the DNA, because to find out the hybridization temperature or welding temperature is the most important part of in the optimization of PCR run, because if you increase temperature once you decrease the temperature, so if you do not decrease the hybridization cannot happen. This is one clause. Another point: if you decrease the temperature, then your target will also get also bind with each other. That means renature back. So you will not find enough number of single stranded DNA. So choosing a hybridization temperature is the most critical step. That's why the normal formula is followed, that we try to calculate the TM of all samples, that is a melting temperature. In melting temperature, it is a temperature where 50% of your DNA present in single stranded form and 50% of DNA present in the double stranded form. In that temperature or annealing temperature should be maintained closer to TM. Now, once the annealing happens, then you use we have tag polymerase in the reaction mix, and it adds the proper DNTP into the strands, and synthesis completes the synthesis of the second strand. So after synthesis of one strand, we have started with one. If we have started with only one copy of the DNA, so after the first round of amplification, we will have two double-stranded copies of DNA. This way, it should happen like a 2 to the power n type of amplification. And if I plot it, it looks like this. Then initial denaturation, next the cycling process, which happens from certain number of cycles, and then the final end of it can hold in the machines. So if the growth is exponential, as I already told and Madam also told, the copy number is 2 to the power n ideally, but in practically we do not find this thing. Why we do not find this thing, and how this thing are find, and what position it is find? We will see exact data of this thing if we see the real-time PCR. So in conventional PCR can tell us what? So we have already seen the conventional PCR can tell us what. That means some gene is present or not absent, or if some sequence is present or absent, we can detect that thing. If the primer probe of that particular gene can bind, if we make a absolute proper primer probe, if it finds the target, then we know something is present. So conventional PCR is basically in qualitative in nature. But if we want to do real-time PCR not only can tell us something is present or absent, real-time PCR can also tell us if something is present, what is the quantity. That means how much it is present. So real-time PCR can be used real-time PCR machine can plot the standard curve into the machine, and based on the standard curve, the quantification of the unknown sample can be done. So this quantification in real time is obviously the relative quantification. If absolute quantification has to be done, it can be done with a variant of real-time PCR, which is known as digital droplet PCR, which is a droplet counting technology, and that is absolute count. But in conventional real-time PCR, whatever chemistry you may use, this basically the quantification is always a relative quantification. That means the quantification which we do for unknown sample are based on the standard curve from which we can obtain, which can be obtained from the standards, given standards. Now, how does real-time PCR work? To understand this thing, let us imagine that we are at the PCR reaction tube at the cycle number 25. So it's just a name, just a 25 is an arbitrary number. At cycle number 25, suppose this is the PCR tube, we all can see. Suppose we are here. Now in the tube at cycle number 25, what can be there? It would be a soup of nucleotide, because some nucleotide would be there, DNTP mix, so primers would be there, template would be there, there will be some amplicons, that means the amplified DNA product, and the enzymes. So it should be present. So if we see, If suppose we have 10 lakh copies of the amplicons at 25th cycle, it's not that 10 lakh copies will be present, it's just an arbitrary number just for our understandings. Let's say 10 lakh copies of amplicon present at cycle number 25. Then using the analogy of the amplicon in every cycle, then assume that at cycle number 25 we have 10 lakh having 2,50,000, we will be having 1,25,000 copies. If PCR happens, PCR doubles the copy number or amplicons in every cycle, or cycle really happens 2 to the power n. Now, what we have done, we have just plotted those data in this curve. So if we plot this data in the curve, and if we keep on doing the 2 to the power n calculation, the curve typically looks like this. And I think those who have seen the real-time data, they all have experienced this thing. That means up to starting from 0 cycle or first cycle, up to a cycle, say 20, 15, 16, 17, 18 here, or it may be extended also in some cases. So up to a certain number of cycles, the curve is going to flat. That means as if there is no amplification. And after that, we suddenly see a exponential amplification, and that will also be there only for few cycles, 3, 4, 5 cycles. And after that also, we will see a flat curve. So after that also, we will see a flat curve. So the amplification or exponential amplification, we can only see for a few cycles. So before that, the curve was coming like a flat, and after that it start amplifying, and the amplification persists only for few number of cycles, and after that it is also going a flat. So it's called the sigmoid shape or S-shaped curve, and typically this shaped curve are called as a complete or good PCR reaction. Why this happened? We'll try to understand this thing. The reason we have started understanding this thing, that initial phases of cycling, the amplification is happening, but the amount of amplification, the amount of doubling is so less that it is not visible in the scale. Suppose from 1 to second cycle, from 1 copy in the second cycle, there will be 2 copies. From 2 copies in the third cycle, it will be 4 copies. From 4 copies, there will be 8 copies, 8 to 16. So if we see the increment is 2, 4, 8, 16, this line. But if we say cycle number 25, the copy number is 10 lakhs and it is absolutely doubling, then in cycle number 26, it will be 20 lakhs. The increment is 10 lakh, and before the increment is 5 lakh, before that the increment is 2.5 lakhs. So the increment is so huge that it is easy. But so at cycle 27 will be 40 lakhs. So we keep on, next another question can come, if we keep on increasing the cycle number, will the copy number will keep on increasing? The answer is no, because after a certain number ingredients which we have given, like the DNTPs and all those other things, it will be exhausted. There will be no more things left. This is the number one reason. And number two reason is probability, because you can understand if we doing the mark to the temp. One template to is record stand, number of copies of DNA present, then the probability will be very higher. So for the probability of finding the primer to the template will be very less. Once it is very less, then the amplification plateau after a amplification. after a number of cycles, certain number of cycles, say 2 to 3 cycles, we really see the exponential amplification. After that, the curve comes at a plateau. That means in those phases, 2 to the power n, whatever we have calculated or have seen in a theoretical perspective, in those cycles, only we can see this exponential. Realistically, the chain reaction progresses, it gets exponentially, it gets exponentially harder to find the primers and nucleotides, which already and the polymerase is wearing out. So exponential growth is not forever. I have already explained this. So after a certain number of cycles, we see the curve will come to a plateau like this thing. Now I am going to the second phase where I told that by using real-time PCR, we can not only detect a DNA or a particular DNA, we can also measure the quantity. So how we can move into that measuring quantity? Let's understand this thing. For understanding this thing, let's use the previous example. Let's imagine that you have start with 4 times less DNA as I do. Then what happened? So as per the previous analogy, I have 10 lakh copies at cycle number 25. So if you have using 4 times less DNA that I do, then you would be having you would be having 4 times more DNA at cycle number 25. That means you would be having 40 lakh copies. So if I get back and use the analogy of 2 to the power n or exponential amplification, that cycle number 24, I would be having 5 lakh. Cycle number 23, I would be having 2,50,000. And for myself, and if I see yours, you are having 40 lakhs at cycle number 25. That means you'd be having 20 lakhs cycle number 24 and having 10 lakhs cycle number 23. That means where I am at cycle number 25, you would reach to the same scale at cycle number 23, right? So let's put it into the graph, and if I put those things in the graph, and the curve and the graph looks like this. So for me, I suppose 10 lakh. Suppose for me, I have put a 10 lakh as a threshold value. Suppose this is our threshold. So I have reached to this threshold or a benchmark at cycle number 25. But you have reached to this benchmark or threshold 2 cycles earlier. Why you have reached 2 cycles earlier to this threshold? Because you have started with 4 times more DNA than what I have started. So now we can understand that by using this thing, I am trying to come into a kind of calculations or quantification. So if you have started, see another example. If you have started with 8 times less DNA, what I have did, then what would happen? Suppose I have 10 lakh copies at cycle number 25 using the same analogy. Then you would be having the 4 times less DNA at cycle number 25. That is 1,25,000. So in cycle number 26, you have 2,50,000. In cycle number 27, you have 5 lakhs. In cycle number 28, you have 10 lakhs. So what I am at cycle number 25, you would take 1, 2, 3 cycles more to reach to that same level if you have started with 8 times less DNA. So even if I put the same data into the curve, it looks like this. Again, our benchmark is 10 lakh. So at 10 lakh, someone has started with 4 times more DNA, reached 2 cycles earlier, and someone who has started with 8 times less DNA has taken 3 cycles more to reach the same threshold. Now, if I consider these samples as standards or something of known concentration, then I can plot this data into a curve and get and make the standard curve, because we already know the quantity or CT value or the cycle threshold value. Now, if I put this CT data with the log quantity, I will be having a standard curve. Now, using the standard curve, if I get the if I have a concentration of unknown, suppose falling here at this position, then I can draw a perpendicular and try and get the concentration of the unknown samples. So this is how usually the concentration in real-time PCR is measured, and real-time PCR can be used this as a quantitative tool, or measuring quantities. It can be used for measuring quantities of viral load. It can be used for measuring quantities of any other mRNA, like for BCR ABL, we measure the quantities of ABL1 copy and the BCR ABL transcript number, and using that we can understand the progression of the disease. So, but you must remember that this quantification, whatever we are doing, that is based on standards, and that is basically a relative quantification. That means the quantification data, so that is why the standard should run well and all the other parameters should be proper, and then only your quantification is valid. Now, Madam has already explained R square, Y, and slope of these things, so I am not going into it. But I can try to show you, try to have your attention on certain point there. If you see this curve, if you see the concentration, this is the log of copy number, and this is the CT value. So even the one copy can be, a single copy can be detected by this technique. That means I try to show you how sensitive this technique is. Now disadvantages of conventional PCR over the real-time PCR. In conventional PCR, you have seen conventional PCR, it is less precision, sensitivity is much low, because you need a difference in the amplicons, because if the two amplicon size is very close. In conventional PCR, those you will see as a single band. It doesn't have a size difference. Then the short dynamic range, only 2 logs can be differentiated, low resolution, non-automated conventional PCR, but real-time PCR, all these are there, all facilities are there. So how does real-time PCR data look like, and what are the MELT curves? So I am just not going details into it, because detailing will take much time, but I'm just giving you a brief idea about MELT curve. So we have seen about the real-time data, and we also have seen how real-time data can be used for measuring the quantities, and how real-time PCR can be used as a quantitative tool. Now, MELT curve is basically the fluorescence data collected and PCR in how much. Now, if you see, if we have using a cybergreen type of chemistry, was Madam was telling, cybergreen of chemistry, we do not have the the targets are not specific. That means if the non-specific targets are also get amplified, and we will also be having the signals for the non-specific target also. So to really know what are there present in our PCR product, we have to do a kind of thing which is called MELT curve analysis. This is the software should be attached to your machines, and the machine have this facility that you can do a HRF belting or high resolution MELT curve analysis. If your machine have that facility, you can do that. Here, what happens? If the same product having a difference, you can be understand. So once you are seeing this data, once if you see the PCR data, raw fluorescence data, all this would be look like this. All should be look like this. But once I convert to a MELT curve, you can see the difference in the population. So there are two population of PCR product are there. To understand this thing, this MELT curve is basically done. Once I convert it to here, it show this is one population of sample, this is one population of sample. So these are application where you to do the genotyping. Suppose HCV genotyping, there are many kits which are based on the MELT curve analysis. Why MELT curve is required? Because between different stains, the sequence similarity is very much there. So if we don't do MELT curve, we cannot differentiate between the two. Now, how resolution MELT curve data different organism is collected, then the data is normalized, then it is compared with each other, and then we can know what different organisms are present. Now, how to optimize the real-time PCR data and troubleshoot problem? Let's see. Optimization is a very important step, because even if you purchase commercial kit, you need to optimize that kit or the data into your machine. Because without optimization, any assay cannot run well in your lab. So a well-optimized reaction will have the evenly spaced standard curves with tight replicates. This is a very important word. So if I make replicates, all replicates should come as a single reaction. So but like here, you can see all the replicates come as a single reaction. It is showing as a single, as if it is a single run. And if I do a dilutions, it should have evenly spaced. That means all should have even space between each other, not more or not less. At 100% efficiency, 10-fold serial dilution should be evenly spaced by 3.3 cycles, ideally. Now optimization, how optimization follows, this is not of a lab part. If lab is using a commercial kit, if the lab is designing the kit, then it's obviously an important part of the lab, that how you have to design multiple primer set. We have to see how the primer set is binding, and then have to assay the optimization. But normally the commercial kits are pre-optimized by the manufacturer, and a typical real-time curve data should have the following characteristic. Number one, S-shaped. Why S-shaped? I have already explained. Because starting from a first cycle to up to certain number of cycle, there will be almost no amplification as per the graph. There will be amplification, it will not be shown. So after that thing, it will keep on doing the exponential amplification, and the exponential amplification will also be valid for only few number of cycles. After that, again the curve will go into a plateau. So if it's typically look like a S-shaped or sigmoid shape like this. And if first S shape, next the dilution series are expected spacing. So dilution series, if we make a 10-fold dilution, it should have exactly 3.3 CT difference. If not 3.3, at least 3.3 plus minus 0.1 or 3.2 to 3.4, then you can know it's a very good dilutions are being maintained. If I make the replicates, the replicate should be tightly clustered, not going to different places. Next, another important, the baseline should be relatively flat, not curvy. Next, if I do a melt curve analysis, then all should come as a single peak. That means only single product is targeted, and that is amplified to ensure. And the curve should be smooth, not the spiky. Next, finally another part, which is the plateau height. The plateau height doesn't matter. That is the amount of fluorophore present and the fluorescence signal emitted by the fluorophore. So it varies from one machine to another. And if you using a Quiescent machine, it also depends on a function called gain. If you increase the gain, then you will see the more height. If you decrease the gain, you will see less height. So plateau height doesn't matter. And if the replicates are not tightly clustered, like if you see here, the difference is something, here the difference is something, here the difference is something, so that it is not properly clustered. And if you see it is dispersed also over the place. So the first reason can be poor pipetting. Next reason can be poorly optimized reaction. This reaction may not be optimized. Then the sample evaporation can also happen, though sealing is not proper. Then the unknown outside range of detection. So it may be the outside range of detection.
Dr. Tridib, Ganesh here, Dr. Tridib.
Detection are not flat. Then the first reason you can see that sample evaporation is happening. Then there may be bubbles. There can be reagents
am I audible?
not poured properly. Yes, you're audible.
Yes, you're audible,
Yes.
Mr. Tanishq.
Dr. Tridib, we are quite late with this presentation. I would request you to wrap it up in the next.
Just within 2 minutes. Within 2 minutes, I'll come. Thank you. And baseline window are not properly set. Next, the dilution series. If you see the dilutions are properly made, it should be like this. If it is not properly made, if something is stretched out or something is squeezed out, then it is not proper. Next, the typical H-sharp shape curve are not maintained. You can see it should be like this, but if it is the curve is like this, it can be a fluorescence drift of unamplified product or something seriously wrong with the experiment. The curve should be smooth, but if you see these kinds of curve, that means the poor pipetting or sample evaporation. I think it is surely sample evaporation. Some serious flaw is happening. And if you see a multiple peak in the melt cup analysis, that means more than one type of product is present. Against is possibly the primer dimers or your primer which are making their self, they are binding with each other, and so it is a problem. The primer needs to be redesigned. And finally, the take-home message is the care in the pipetting and care the choice of the plastics and sealing plate properly. There should not be bubble present. Then you should see ultimately your curve should be like this. Thank you.